Hello, hello. Welcome back. Chapter 7 today. So this is a long chapter. Again, we're probably going to have to break this up into a few videos so that we can get all the content in. And I got a new toy from IT. With all the new modifications on my machine, I have a pen. So I'm going to try playing with this pen to do highlighting and stuff. We'll see how it works. All right, today we are going to start on gene linkage and mapping. So one of the things that is kind of interesting about genetics is, especially after you look at Mendel's laws. One of Mendel's laws identifies whether or not two genes kind of travel together, and that's where he was doing those dihybrid crosses. You know, the idea was that two genes with different alleles would be transmitted independently of one another. Now, it turns out that that is often true unless two genes travel on the same chromosome. So if you have two genes that travel on chromosome 1, it turns out that sometimes you'll get a higher proportion of offspring demonstrating those traits. So that's what we call, in general, gene linkage. And so that's what we're going to be kind of discussing today. So as it says here, each species of organism has hundreds of thousands of genes. And of those hundreds of thousands of genes, there are many alleles for each gene, 2 or more. But every species only has a few chromosomes. So each chromosome itself carries hundreds of genes on it. And so since these genes on that single chromosome would be physically linked to one another, then we call those gene linkage groups. So, okay, my first mistake there with the marker. We'll see how this goes. So Bateson and Punnett discovered that two traits don't necessarily always sort independently, doing those dihybrid crosses, or in general, getting that 9:3 to 3:1 phenotypic ratio in the F2 generation when you're performing dihybrid crosses. And they discovered this looking for genes involving flower color and pollen shape. And so when we look at this particular transmission pattern, there are Ps that can be purple, pea flowers that are red, and then there are long pollen grains or round pollen grains. And so it turns out that purple is the dominant color over red, and long pollen grains are dominant over round. When you do a dihybrid cross, doing homozygous dominant versus homozygous recessive for both traits, you'll end up with a heterozygote that looks intermediate, well, not even intermediate, I shouldn't say that, but that has the appearance of the dominant trait. However, the F2 offspring, when you look at that, you do get a greater proportion of the two parental genotypes here, purple flowers, long pollen, red flowers, round pollen, okay? It does sort 9:3 to 3:1 in this particular case. You do get, and these are the actual counts. However, when you do the calculation, you have 15.6, 4.5, which is a little off. So it turns out that when you get these slight variances, when it's not so much 9:3 to 3:1, but let's go back up here, more like 15:1 to 1 to 4.5. I mean, you know, if you count more, you will end up getting, the math will end up proving out. But it turns out that these genes appear to be what we call coupled, or they're transmitted together because they travel on the same chromosome. When we have chromosomal transmission, those are called linkage groups. Linkage groups are groups of genes that, for example, will travel together. So in humans, we have what we call 22 autosomal linkage groups. 22 autosomal groups have to do with having 22 individual chromosomes 1 through 22. Then you have the X linkage group and the Y. So you have 22, 23, for the X and Y, the sex chromosomes. Now, 23 plus 23 is 46. Right now, we'll focus on haploid number, okay? Haploid meaning half. So 22 autosomal groups, so chromosomes 1 through 22, only one of each, and then either the X or the Y. So it turns out that sometimes these genes can be far apart on a chromosome. So let's look at my pen here, if I can get it in the field. Let's say, for example, you look at the pen and the top and the bottom, as you can tell, are fairly distinct from one another. If you have a gene near the bottom and a gene near the top, these two genes will tend to get mixed up from one another pretty easily. But if you have two genes sitting right next to each other, it's difficult to separate those genes from each other because of crossing over. What is she talking about? Oh my gosh. All right. It turns out that independent assortment works if genes are far away from one another. So Mendel's assumptions worked really well for genes that were on different chromosomes. Genes that are on the same chromosome, on the other hand, have some issues in terms of how often you can split them up from one another during recombination or crossing over. So when you're doing a dihybrid cross, during dihybrid cross studies, and you're looking at crossing over between two genes, sometimes if the two genes are close together, again, if you have them close together like here and here, it's difficult for them to cross over and mix up. But if they're far apart, crossing over is occurs more easily. Let's review real quickly. Crossing over occurs during prophase 1 of meiosis, and that's after sister chromatids have replicated. Remember, the two sister chromatids are sitting right next to each other, and then boop, you have a crossing over event occur. Non-sister chromatids are not going to exchange DNA segments, so you are not going to see, hopefully, unless a mistake happens, which sometimes does happen, you are not going to see crossing over, let's say, between chromosome 1 and chromosome 3. You'll only be seeing crossing over between chromosome 1 or sister chromosome 1 and sister chromatid 1 together. This is a better picture, a better idea of what's going on rather than using my fingers, right? So let's say over here, this cell has already gone through S phase and G2 phase. All the DNA has been checked for spelling errors, and now the cell's going to undergo meiosis. Here you have mom's chromatid pair and dad's chromatid pair, okay? Remember, chromatids are just duplicated chromosomes. This is one chromosome from mom and one chromosome from mom held together at the centromere. And here's dad's. Mom is carrying dominant versions of each gene, so dominant alleles, and dad is carrying recessive alleles. Because of meiosis, if no crossing over happens, mom transmits one chromosome to one gamete, and dad will transmit his to individual gametes. So basically over here, without crossing over, it appears as if every allele sorts independently. You have independent segregation because you didn't have mixing. However, let's come here to the next slide. If you had crossing over occur, these two sister chromatid pairs look like they're kind of, kicking each other. Then you'll have mixing of the terminal ends here. So big A and big B is going to get swapped around. So over here, this first line here, this first chromosome right here, okay, my pen's not working, sorry about that. This first region here is going to be transmitted here. So we're going to make this number 1. This is chromatid, or it turns into chromosome 1, that gets separated into this cell. This one right here gets sent on over to this possible haploid outcome. Now, this region right here is going to mix or cross over, meaning that this side goes over here and this side goes over here. So now you have this going there and this going there. So you have a mixing of chromosomal content. And so this is what we call recombination. These two, cells 1 and 2, or gametes number 1 and 2, are non-recombinants. Sorry, I have terrible handwriting. I'll try to do better. These two are recombinant gametes. A-N-T-S, sorry. These are recombinant gametes. And what this means now is that these two, gametes 3 and 4, are going to give rise to new combinations. So if you had big A being brown and little a being white, let's say for fur color in mice, and big B and little b being, let's say big B being black eyes and little b being red eyes. Okay, so A is fur color and B is eye color. Now what you have is this mouse will have mouse number 1, or at least this outcome, will give you brown fur and black eyes. Number 2 is going to give you white fur and red eyes. Over here now, and this is what the parents originally looked like. Over here, you're going to see black eyes and white fur. And over here, in this last quadrant from gamete 4, it's passing on red eyes and brown fur. Freaky mouse, right? Crazy looking mouse. So the idea is, by crossing over occurring, you're mixing up chromosomes. You're mixing up allele representations. And by that occurring, you're going to end up with new variances or new combinations in the offspring. So that is what we call, let's come back here one more time, if they're not separated, we call those linked genes. Or in this case, they're genes because we're only dealing with genes, not necessarily individual alleles. They can be linked alleles as well. If they're far apart, they may be not tightly linked. If they're close together, the linkage would be very tight. If you have a gene, let's say A and B, genes A and B are a linkage group, which we'll run into in a minute, so I'm not trying to throw too much at you yet. But now let's say we introduce gene C. Gene C is going to be on a separate chromosome. Therefore, it's not linked to gene A and B. So Morgan himself produced some ideas and evidence that linkage occurs very strongly on the X chromosome. So we're going to start looking at X-linked genes. In certain organisms, there's an X-linked pattern of inheritance. Now Morgan, Thomas Hunt Morgan, was a researcher that worked on Drosophila. And one of the beauties of Drosophila, or fruit flies, is that they have a small chromosome number. Females do have 2 X chromosomes. So what Morgan was able to do was look at body color, eye color, and wing length. All 3 traits are X-linked traits that are transmitted to offspring from the parents. All right, so this looks kind of scary, I know. Here's a female. This symbol means female, and this symbol means male. The females are always larger than the males. Females have 2 X chromosomes, hence XX, and males have XY. XY. Now, in Drosophila, one of the things that Morgan started doing, instead of doing big A, big B, little b, and so forth, Morgan started using a different annotation. And so what you see here, YWM, means yellow body, white eyes, and M is for miniature wings. Mini wings. Okay, each one of these traits are going to be the recessive. So yellow, white, mini. The dominant, all right, so you just have Y for yellow, W for white, M for mini. These are recessive. Okay. Now, over here, Y plus is red. What does that mean? Y plus is what we call the wild type phenotype, the red eye. So if you look at flies, most flies have eyes that are kind of a reddish color. W plus is actually a black or gray body. W plus is that ugly black fly body. If you love flies, I'm sorry, I don't particularly care for flies, but black body is wild type. It's normal. And then M plus equals normal wings, or normal length wings, okay? Normal wing length. All right, so mini wings, eyes, yellow body. These are recessive. Red eyes, black body, normal wing length. These are the dominant. So in this particular case, you're looking at a different annotation that can look confusing. After a homozygous recessive female was crossed with a homozygous dominant male, okay, the reason why, actually it's not even homozygous, it's X-linked. My apologies, X-linked. You end up with females that look different because the female is now been given a copy of the dominant traits from dad. The males have received, half of them are going to receive mom's chromosomal composition. Now, we'll work out the rest of this on the next video.